In this session, I'll be discussing about one important aspect of amino acid metabolism, that is 1-carbon metabolism. I'll be discussing in two parts. The part 1 is about how the 1-carbon groups are generated and how they are interconverted. Okay. So at the end of the session, the learner shall be able to describe definition of 1-carbon groups, 1-carbon, different 1-carbon groups, generation of 1-carbon groups, interconversion of 1-carbon group from one group to other group. Okay. So let us first we'll talk about some general aspects of 1-carbon. This one groups. Let me define it. Okay. So when you talk about the synthetic, it is synthetic, or we can say anabolic pathways. Or there are metabolism can be anabolic and catabolism in the synthetic or anabolic. Okay, pathways. These pathways require what they require. So they require addition of 1-carbon groups. One of the aspects of synthetic or anabolic pathways require addition of 1-carbon groups. Okay, so when you talk about these 1-carbon groups here, so as I said, this is one important aspect of amino acid metabolism. Amino acid metabolism, known for many important aspects. So on many counts, it is important. So let us see, in one aspect, the amino acid metabolism is important, is the transfer. One is transfer, or we can say transfer or exchange of. Exchange of 1-carbon groups, 1-carbon units. We can say 1-carbon units. So for this, amino acid metabolism is an important aspect, one of the important aspects. Now, let us first take up the first objective. In the first objective, we'll define the 1-carbon. What is 1-carbon? Okay, so group. We'll define it. What is 1-carbon group? So when you talk about the 1-carbon group, an organic molecule, molecule in a simple terms, having organic molecule having a single carbon atom. It is as simple as that, a single carbon atom. So organic molecule having single carbon atom, so that we can include in the 1-carbon group. So now there are number of 1-carbon groups are there. Next objective, the second objective which I will take is categorization of 1-carbon groups. So as I said, there are many 1-carbon group, many 1-carbon groups are there. So how they are placed, how they are categorized. So what is the basis of that categorization? The basis which I have taken is, in terms of redox state, which one is most oxidized and which one is most reduced, what is in intermediate level. Okay, so there are three aspects, I will keep it here. So that is group, name, group, and redox state. Okay, so the first one which I will take is carbon dioxide. Here, carbon dioxide, that is the most oxidized. So one, the first one which I will take is the carbon dioxide in this name and group and redox state. So the carbon dioxide, we say it is CO2. The second one is a formyl group. So it is formyl group. So that can be represented as CHO. Then the next one is form amino. That is form amino. That is CHNH. Okay. The next one is methynyl. So here it is CH. Okay. These four I'll be placing in most oxidized. Of course, carbon dioxide is the most oxidized, followed by others. So I can say they are most oxidized. So 1-carbon groups. Next, I will take up the intermediate level, and then most reduced I will take up. So here in the intermediate level, hydroxy, methyl, that is the one. There are two are there, and also methylene. Okay, hydroxymethyl is represented, formula is CH2OH, and then methylene is CH2. Okay, then it is in intermediate stage, okay, redox state. Finally, the most reduced, which I will talk about, is methyl. It is, we can say, methyl. Here it is CH3, and it is most reduced. Of course, there is another one is there, the most reduced, that is methane. Next, here we have this CO2. First, I will talk about the CO2. What is CO2? CO2 is dehydrated form of carbonic acid. It is dehydrated form of carbonic acid. Okay. Then one thing is there, it is not considered. It is not considered in one carbon pool. Okay. When you see the most of the books, it is not considered in the one carbon pool. Okay. Then what I do is next, I will categorize the groups into most oxidized and most reduced among these. So one carbon, the groups I will categorize into most oxidized, most reduced. Most categorized is CO2. So CO2 is handled, if it is not included in the one carbon pool, who handles the CO2? It is handled by thymine. Okay, pyridoxine, and biotin. So here the thymine and pyridoxine, thymine is mostly carbohydrate metabolism, pyridoxine is amino acid metabolism, and biotin is actually involved in the carbon dioxide fixation. So we can say in the gluconeogenesis, its role is there, and its role is there from the conversion of propanol COA to methylmalonyl COA. Okay, and also its role is there in fatty acid synthesis. Also, its role is there in the branched chain amino acid metabolism. Okay, then most reduced is methane. Methane is not important. It is not important. Okay, important. So in human beings, it is not important in human beings. Okay, so that is the most reduced and most oxidized. Then next we'll take up the third objective. What is the third objective? The generation of one carbon groups. The third objective is how these one carbon groups are generated. So let us study about this aspect. The first one in the generation of one carbon groups, we'll study one by one. A is conversion of serine to glycine. There is glycine formation from the serine. Okay, let us say you take the serine is hydroxyl group containing amino acid. It is one of the amino acid which contains hydroxyl group. Other amino acids threonine and tyrosine. This is serine. Okay, serine by the action of enzyme serine hydroxymethyl transferase, that is we call it SHMT. So here what happens, the methyl, that is group is taken. So methylene group is taken by the folic acid coenzyme, that is tetrahydrofolic acid, leads to formation of N5, N10. Okay, so methylene, okay, THFA. So what it is formed? It is formed glycine. It is glycine. Okay, so here glycine formation has taken place. It is called glycine. Simplest amino acid glycine formation has taken place. That is the one reaction where the one carbon group is generated. The next reaction here, the B. In this one, okay, then the glycine cleavage system. What happens to this glycine cleavage system? Okay, here you can say this is glycine. Okay, the glycine, this is glycine amino acid. Okay, then this amino acid, glycine amino acid, this is a reversible reaction. Above also, when we talk about the serine, so that is also reversible reaction. Okay, so they can be interconverted. Glycine here. So there is a glycine cleavage system is there. Glycine cleavage system is there. Okay, so in the glycine cleavage system, okay, then again you see, so here, here the glycine cleavage system is there. Just arrow I have to change. Okay. here it is, then it is, okay, THFA, the THFA is converted to, okay, N5, N10, methylene, N10 and N10, methylene, THFA, N10, N5 and 10 methylene THFA leads to formation of CO2 plus, okay, NH4. Okay. Then the third reaction is in amino acid metabolism, that is tryptophan. Tryptophan, amino acid, it is an aromatic amino acid, okay, heterocyclic amino acid. So tryptophan, in one of the steps, it is converted to formylkynurenine. It is one of the steps, it is converted to the formylkynurenine. So that means it contains a formyl group. It contains a formyl group. Okay, then here, so there is to the NH, formyl group is attached. So this is called formylkynurenine. Okay, this formylkynurenine, by the action of form amidase, okay, form amidase, okay, amidase, then what happens? The formate group is released. So the formate group is released, okay, here in the presence of H2O, the release of formate group, okay, formate group is released, that goes to the 1 carbon pool. That goes to the 1 carbon pool. Okay, leads to formation of a kynurenine. The next one is, so in this category, the last one, okay, histidine. Histidine, through many reactions, okay, number of reactions, is converted to form aminoglutamic acid. Form amino, so okay, so converted to form aminoglutamic acid. Okay, here you can see, so form amino group is there, then a glutamic acid, okay, then, okay, this is called form amino, okay, glutamic acid. It is form amino glutamic acid. Okay, then this is also in short form, we call it FIGLU. Okay, then what happens? This FIGLU, what is the fate of this FIGLU? Okay, here what happens in the presence of THFA? In the presence of THFA, it is converted to N10. N10 form amino THFA. N10 form amino THFA. So leading to the formation of glutamic acid. Okay, glutamic leads to formation of glutamic acid. Okay, here you can see the glutamic acid is released. The glutamic acid is released. Here it is called glutamic acid. So see, if THFA is not there, what will happen? Form amino glutamic acid cannot be converted to glutamic acid. Then this level of FIGLU level will increase in the absence of the THFA. That means in the deficiency of folic acid, the levels of the form aminoglutamic acid increases in the urine. There increased excretion of form aminoglutamic acid in the urine. So that is the reason it is used one of the tests to detect the deficiency of folic acid. Okay, this is how the generation of 1 carbon groups is taking place. How the generation of 1 carbon groups is taking place. Next objective is interconversion. Interconversion. Okay, that means conversion of 1 carbon group to other carbon groups. Interconversion of 1 carbon groups. 1 carbon groups. That is the next objective. Let us see. First, we'll start with the folic acid. We'll start with the folic acid. Let us see the structure of the folic acid. Okay, folic acid contains a stearoic acid. Folic acid contains a stearoic acid. Okay, then, okay, here like that. So here there is a OH group. Here it is NH2. Okay, then here, okay, then, so here H will be there. Okay, then there is para-aminobenzoic acid. There is a presence of para-aminobenzoic acid and also glutamic acid. The glutamic acid number may change. Okay, let us see the numbering of this one. That is important for us to know about this aspect. Okay, then here it is 5, 6, 7, 8, and okay, 9, 10. Okay, so what we are interested is, so this is tetrahydrofolic acid. This is tetrahydrofolic acid. So what we are interested in this one is about this aspect, this one, this nitrogen N5, N10, where the 1 carbon groups are attached, different 1 carbon groups are attached. Okay, then let us see here in this one, in the post reaction. In the post reaction, what happens? In the post reaction, in the presence of formate, here in the presence of formate. So in the presence of formate, what happens? Leads to formation of N10 formyl THFA. This is called N10 formyl THFA. See the way this is the 10, this is 5, where the group is attached here. What happens? So there is dehydration taking place, H2O is released, then again it can be H2O added reverse reaction. Okay, then what happens here? Let us see. In this one, there is, okay, here you can see, okay, then here it is. Then again, you say, here it is, okay, so here, again you say, this is 5, this is 10. What we call it, both 5 and 10 are involved. This is we call N5, N10 methinyl. There is a double bond is there, methinyl. Okay, N5, N10. So methinyl THFA, N5, N10. So methinyl, you can say methinyl THFA. Okay, so here it is. Then what happens to this one? Here, again, this can also be, okay, reversible reaction, where, so there is a presence of dehydrogenase, in the presence of dehydrogenase, okay, dehydrogenase, NADPH plus H plus leads to NADP plus. Then what happens here? You can see here, CH2, here it is again 5, here it is 10. Let me again show you how it is taking place. So what we call it, this one? So we call it, okay, N5, okay, N10, methylene, N5, okay, N5, N10, methylene, that is methinyl, okay, methylene, THFA, okay. Then finally, what happens? There is one very important reaction which is taking place here. Okay, one important reaction which is taking place, that is an irreversible reaction. So again, I am writing the N5, this is the THFA, okay, this is the, here it is, THFA, okay, let us see, okay, N, here it is, okay, glutamic acid, then, okay, here it is, okay. Then what happens here? So it is an irreversible reaction is taking place. Irreversible reaction is taking place. What is that reaction? Where the methyl group is, there is a formation of N5 methyl THFA, N5, which is called N5 methyl, okay, THFA, N5 methyl THFA. So here it is an irreversible reaction. So you can say it is an irreversible reaction. In this irreversible reaction, see, you can see this is the irreversible reaction. Here, which one is participating? Here it is reductase enzyme. Reductase enzyme is participating, where the NADH plus H plus is converted to NAD plus. Here you can see here in this one, okay, here see. So like this is N5, it is N10. N5 methyl THFA, how the interconversion is taking place. So when you go back here, the first one is, so the formate group is added, leads to N10 formyl THFA. Then the next one is methinyl THFA, N5, N10, and the next one is methylene THFA, the next one is N5-methyl THFA. This reaction is irreversible. In interconversion, once the folate is in the form of N5-methyl THFA, it cannot go back. It requires to transfer its methyl group the presence of B12. That we'll study in the next session. Okay, about the B12. Okay, then there are other aspects are there. The interconversion, there are other aspects are there. What are those other aspects? Suppose this N5, N10, okay, methylene THFA. We have the methylene THFA. So as I said, so that is in the oxidation. If you carry out the oxidation, so there will be formation of N5, N10, methynyl THFA. Methynyl THFA. This methynyl THFA. So this methynyl THFA, okay, so have the, it can be, give the, either it can give the N5 formyl THFA, also it can give the N10 formyl THFA. N5 form amino THFA. Then what will happen if we remove this NH3 group? It can give the N5 N10 methynyl THFA. Okay, so this is the another form of interconversion. Then we have the N10. So if we have the N10 hydroxymethyl THFA, so this also can be interconverted. So into N5 N10 methylene, okay, THFA. So these are the various interconversions. With this we have studied or completed all the 4 objectives. Let us summarize 1-carbon metabolism. Okay, in the part 1 of 1-carbon metabolism, the objectives we set on. At the end of the session, the learner shall be able to describe definition of 1-carbon group, different 1-carbon groups, and generation of 1-carbon groups, and interconversion of 1-carbon groups. The synthetic and anabolic pathways require an addition of 1-carbon groups. Okay, synthetic pathways or anabolic pathways. Amino acid metabolism is very important in many aspects. One of the importance of amino acid metabolism is transfer or exchange of 1-carbon units. Now, in the first objective, we have studied about the definition, where, so we said an organic molecule having a single carbon atom that can be called 1-carbon group. Then, so we categorized these various 1-carbon groups into different oxidation states. The first one, it is name, group, and redox state. So carbon dioxide, CO2, formyl, CHO, form amino, CHNH, and methynyl, CH. And they are the most oxidized one. Okay, then, so hydroxymethyl, methylene, CH2OH, CH2, in intermediate oxidation state, redox state. Then methyl, CH3, most reduced. Then CO2 is the dehydrated form of carbonic acid, not considered in 1-carbon pole. And we have placed the most oxidized CO2 and most reduced, that is methane. Okay, the most oxidized CO2 is dealt by the thymine coenzyme, pyridoxine in carbohydrate and amino acid metabolism, biotin is in the carbon dioxide fixation reactions, and next, so methane is not important in the human beings. Then we talked about how the 1-carbon groups are generated. Serine to glycine, where serine is, so the transfer of methylene group is taking place, THFA, leads to N5 and 10 methylene THFA, leading to formation of glycine. In the next reaction, so we talked about how the glycine, by the glycine cleavage system, THFA is converted to the N5 and 10 methylene THFA. Then leading to formation of CO2 and NH4. Then we talked about the tryptophan, okay, then aromatic amino acid, and leads to formation of formyl kynurenine. This, by the action of formamidase, leads to formation of kynurenine, releasing formate group, and that goes to the 1-carbon pole. Then we talked about the histidine. Then, so releasing the form aminoglutamic acid, figulum, and THFA and N10 form amino THFA formation has taken place, then leading to the formation of glutamic acid. Then, in the last objective, we talked about the interconversion of 1-carbon groups. We talked about the tetrahydrofolic acid. Okay, then teroic acid, para-aminobenzoic acid, glutamic acid. Two groups are important, N5 and N10, in 1-carbon metabolism. Okay, then in the presence of formate leads to formation of N10 formal THFA, releasing of the water, then leading to the formation of N5N10. Okay, the methynyl THFA, that is by the action of dehydrogenase leading to the formation of N5N10. Okay, methylene THFA. Then this one reductase, that is irreversible reaction. So okay, so NADH is converted to NAD plus, leads to formation of N5 methyl THFA. Then also we talked about the other types of interconversion. Methylene THFA, oxidation to the methynyl THFA, that can be lead to the formation of N5 formal THFA or N10 formal THFA. Okay, then the N5 form amino THFA leads to formation of N5 N10 methynyl THFA. Okay, then also finally we talked about the N10 hydroxymethyl THFA converting to the N5 N10 methylene THFA. This is about the how one can define the 1-carbon and what are the different 1-carbon groups, how they are generated, how they are interconverted. In the next session, we'll discuss about how these 1-carbon groups are utilized. Okay, thank you very much.